screening of blood for infective material. So in the first part we have seen what and all are the transfusion transmissible diseases and their diagnosis. So one of the important tests comes under screening is the ELISA. So this is the first test developed for the detection of HIV antibodies in the year of 1985, and currently it is a widely used test. So there are three methods, that is indirect ELISA, competitive ELISA and sandwich ELISA. In indirect ELISA, antigen is coated to the well and antibodies in the serum is detected using conjugate. Whereas in competitive ELISA, antigen is coated to the well and there is a competition between antibodies of the test sample and the conjugate to bind to that antigen coated to the well. Third one is sandwich ELISA. It is a highly specific method. So here antigen or antibody can be used as a sandwich format. So here we can see antibodies. To detect antibody, it is sandwiched between the conjugate and antigen. Antigen is bound to the microplate. Here for the detection of antigen, antibody is coated to the well, antigen is sandwiched between the conjugate and antibody. So that is about the three methods of ELISA. Moving to the requirements of ELISA, we have the list here: microwells, sample diluent, conjugate, wash buffer, substrate, stop solution, controls, PC and NC, and we have the plate sealer during incubation purpose. So we need to dilute the conjugate, so we have the diluent along with the kit, and for the dilution of substrate also we have the diluent. Stop solution usually used is dilute sulfuric acid. So moving to so here we have the image of ELISA kit along with the microwells and all the reagents here: wash buffer, sample dilute, conjugate diluent and all. So moving to the procedure. So indirect ELISA here I have given is, that is for detection of HIV antibodies. So to detect antibodies, the surface of the wells to be coated with the antigen. So microwells coated with the antigen. Now add the test serum to the well and include the PC and NC. So add the controls to the positive control and also to the negative control. So whatever the procedure we follow for the test to be followed for the controls also. Now after adding the serum, incubate for at 37 degrees Celsius for 30 minutes to 60 minutes. After incubation, washing is done to remove the unbound material. So whatever the antibodies remains without binding is removed by washing method. Now add the conjugate. So conjugate is also known as anti-human antibody, which is linked to the enzyme. So it has the ability to bind to the antigen antibody complex. So now after adding conjugate, again incubate. Incubate at 37 degrees Celsius for 30 minutes. So next again washing is done. So washing procedure, we have the ELISA washer now, or manually also we can do the washing, aspirating and discarding. So that procedure can be done. After washing is done after adding conjugate to remove the excess conjugate or unbound conjugate. Now add substrate. So substrate is nothing but it contains a chromogen. So the enzyme present in the conjugate causes oxidation of this chromogen and results in the color. After adding substrate, again incubate at room temperature for 30 minutes. And after incubation add blocking agent. So blocking agent is dilute sulfuric acid. So this stopping agent, it causes inactivation of the enzyme and also it fixes the color. So finally after adding the blocking agent, now read using ELISA reader. That is OD values are obtained by measuring in the ELISA reader. So moving to the procedure again, antigens are coated to the surface well, add the test serum and the controls, incubation. After incubation do the washing to remove the unbound antibodies, you can say, or unbound material. Now add the conjugate which will bind to the antigen antibody complex and incubate. After incubation, do the washing to remove the excess conjugate. So substrate or the chromogen containing reagent is added. So enzyme present in the conjugate causes oxidation of the oxidizes this substrate solution to develop the color. Now do the incubation and add blocking agent, so which will inactivates the enzyme and fixes the color. So finally the color change is measured in ELISA reader. So here we can see antigen is coated to the well, antibody in the test serum, adding conjugate, so which conjugate will bind to the antigen antibody complex, add the substrate, enzyme oxidizes the substrate to produce colored product. So here also we have the same thing, antigen coated to the well, antibody in the serum, conjugate, that is conjugate will bind to the antigen antibody complex, so substrate is added, enzyme causes oxidation of the substrate and results in the colored product. Moving to the interpretation of the test results. So in the ELISA reader we will get the OD of the each well. So after getting the absorbance value or optical density of the each wells, so we need cutoff value to report the ELISA results. So how to calculate the cutoff value? Each kit provides its own method to calculate the cutoff value. So example is, it is a mean of positive control, mean of the negative control is usually maybe the formula for cutoff value. So using the cutoff value you will get some values here. So when we calculate, if the test absorbance value is above this cutoff value, then it is reactive. If the test absorbance value below the cutoff value, it is negative or non-reactive. So this is how interpretation of the test results are done. So OD values are obtained for each well in the ELISA reader. So now calculate the cutoff value. So cutoff value formula will be provided in the kit itself. So after calculating the cutoff value, now the absorbance value is compared with that cutoff value. Absorbance value above the cutoff value is reported as reactive. Absorbance value, test absorbance value below the cutoff value is reported as non-reactive. So that is about our indirect ELISA. Moving to the next, that is a sandwich ELISA. So here I have given example of sandwich ELISA for detection of hepatitis B surface antigen. So hepatitis B surface antigen, it is the first marker to appear in case of hepatitis B virus infection in the serum. So to detect antigen, the microvelles to be coated with the antibodies. Now add the test serum. After that, incubation. After incubation, washing is done, and now add conjugate which will bind to the antigen antibody complex, and now incubate at 37 degrees Celsius for 60 minutes. Washing is done to remove the unbound material and add the substrate now. So substrate is the coloring or the chromogen containing reagent. So enzyme present in the conjugate oxidizes this chromogen to give a color product. After adding substrate, incubate, and now incubate at room temperature for 30 minutes, and finally add stop solution or blocking agent, that is dilute sulfuric agent, so which inactivates the enzyme and fixes the color, and finally take the reading in the ELISA reader. So that is about the sandwich ELISA. So antibodies are bound to the well. We are adding the antigen present in the test serum, that is hepatitis B surface antigen here. Incubation, washing, adding conjugate, incubation, washing, adding substrate, incubation, adding stop solution, and then reading in the ELISA reader. So here we can see sandwich ELISA. Antibodies are bound to the microvelles, antigens in the test serum, conjugate bound to the antigen antibody complex here, washing to remove the unbound conjugate, so substrate is added, enzyme oxidizes the substrate to produce the colored product. Merits of ELISA. The ELISA is a highly sensitive and specific test. It is economical for large blood banks. That means many tests can be performed at a time. Going to the demerits, here it requires maintenance of the equipment. So we have the equipment like ELISA reader and we have the washer. So it requires well-trained manpower. So here the technologies need to be trained well, and it is not ideal for small blood banks since we need more instruments here. So it is not ideal for the small blood banks. So these are the merits and demerits of ELISA. So indirect ELISA, indirect sandwich ELISA here. Here we have the instruments like ELISA reader. ELISA reader, it will display the OD values of each wells here, and we have the washer. So by this I'll end ELISA, and next class I will explain about particle agglutination assay. So that's all about ELISA.